Okay, our third person in the predation studies is Cynthia LeDoux-Bloom, and she studies the habitat and movement patterns of freshwater, anadromous, and marine fishes using acoustic telemetry and other technologies. She recently has expanded her research interests to investigate habitat use of abalone using ROV and aging sea cucumbers, using ossical morphometrics. She's currently a postdoctoral volunteer with the California Department of Fish and Wildlife. I got it right this time. With the Marine Invertebrate Program in Fort Bragg, and a senior fisheries biologist with AECOM in. She holds a Bachelor of Science in Marine Biology from UC Santa Cruz, a Master's in Fisheries from San Jose State Moss Landing, and a PhD from UC Davis in Animal Biology. Cynthia?
Thank you very much.
Just go ahead and get you fired up here.
Okay. And I'm right there. Perfect. Thank you, sir.
All right. Yeah, that's fine.
Okay, great. Oops. Anyway, thanks again for inviting me to talk today. This is a collaboration of work between myself, Doctors Johnson, Klimley, Serge Doroshoff, and Siki Fan, all from UC Davis, and from Jeff Isley from the Southwest Fishery Center in NOAA. Today I'm going to be talking to you about habitat use, movement, and residence patterns of subadult striped bass. And specifically, as terms of habitat use, I'm going to be focusing on salinity, water temperature, and a little bit with depth in regard to shoal and channel use. I'm not going to have time today to go into the analysis that we did with turbidity. So if you're interested in that, please get in touch with me and I can send you a few slides. This work right now is in its final stages of a manuscript that's going to be submitted by the end of this month to the Journal of Animal Biotelemetry. So before we get started, I really want to acknowledge the people that assisted with this program. Funding was provided by the Department of Water Resources and the Interagency Ecological Program while I was employed by DWR. Funding sources also came from UC Davis's Animal Biology Graduate Group, the Stockton chapter of the California Striped Bass Association. The habitat data was provided by NOAA and the California Data Exchange Center, Reclamation, USGS, and the Central and Northern California Oceanographic Observing Systems. And the telemetry data was provided by the California Fish Tracking Consortium, NOAA Fisheries, the California Department of Fish and Wildlife, Reclamation, the Army Corps of Engineers, Bud Abbott, E-Corp, who else? One other person. E-Corp, Bud Abbott, and Hansen Environmental, and many other people. And last but not least, I couldn't have been able to do this project without 50 volunteers and interns that assisted me. It was a big project. I am very grateful to all the help that I got during this time. So as Louise just finished up, striped bass are not centrarchids. They are true temperate bass in the family Moronidae, and they have a truly plastic life history. They are really the coolest fish on the planet. They're euryhaline, and they're able to inhabit water from fully fresh at 0% parts per thousand all the way up to euryhaline at 33 parts per thousand. And because of their osmoregularity, it's easily for them to go back and forth between differences in salinities, which we do not see with smolts. They're also eurythermal. They inhabit water temperatures from 8 to 28 degrees Celsius, and they're anadromous. In fact, they're so anadromous that otolith studies on the East Coast have shown that subadult striped bass actually spend years at sea. They exhibit bimaturation. 100% of males are mature at age 2, whereas only 25% of females are mature at age 4, 75% at age 5, and by 100% are completed by the time that they're 6 years old. This in itself is pretty problematic in that the California Fish and Wildlife Regulation currently sets a length of 18 inches total length in the fishery. So females don't even begin to mature until they're over 24 inches. So our fishery right now, regulation as it is, completely fishes for females that have never had an opportunity to spawn and are immature, and overly fishes for males that are mature. They also are iteroparous. They can exhibit skip spawning, and they have been known to live up to 30 years. So the life cycle of the striped bass, really quickly, it's pretty complex, but it's not what we're here to talk about, but I wanted to make sure that we all were on the same page. So the adults start moving back into riverine habitat from estuaries and ocean when the water temperatures here in the San Francisco Estuary watershed start approaching 16 degrees Celsius, very similar to on the East Coast. The larvae are transported downstream into the low salinity zone where they hang out until they're juveniles. There is some knowledge of juvenile movement back up into freshwater with the tidal influx and then coming back down to the low salinity zone. And then subadults are thought to have been spread out throughout the watershed over the whole entire year. So knowledge of striped bass in the San Francisco estuary is, we know a lot about them, really. We have all kinds of governmental agencies, both state and federal, that are looking at the egg, the larvae, the juvenile, and the adult stages. The big black boxes are A, subadult striped bass, their habitat use, migration patterns, and their residence, and what the heck is going on in the ocean. So until we have a handle on what is truly going on in the ocean, we're not going to have a good idea about predation on smolts, because they're probably following them out there as they out-migrate and immigrate into the ocean. I'll show you that in a video. So really briefly, I wanted to go over this. Oops, here we go, top one. So this is the area of the San Francisco Estuary watershed that my fish inhabited during our study. So the section down here, here's the Pacific Ocean. This is relatively high salinity. It's euhaline, polyhaline, and during high flows, the section here will decrease into mesohaline. So we're gonna call this just the Bay Area. And this area right here is a low salinity zone, which is what folks call it around here, basically, and it's from meso to ogliohaline. And at this confluence here, this area here is predominantly freshwater. It rarely goes over 0.50 parts per thousand. And so when we've been talking today about salt water coming up here, we gotta kind of remember that salt water from like a Homo sapien perspective, in that the water that we drink. It's not so much salt water in relationship to a fish. And how Louise just so rightly pointed out that we really are gonna need to probably start looking at large and other centrarchid bass habitat down here and look at predation with them. So basically, I'm going to talk to you briefly about the tagging methods. We caught hundreds and hundreds of subadult striped bass. We only selected 99 to implant with acoustic transmitters. I conducted all the surgeries myself. The V9 acoustic transmitters were surgically implanted into their interperitoneal cavity. I quickly closed the suture with 3 interrupted, or the incision with 3 interrupted sutures, and we released all the fish at the site in which they were captured. So another really cool thing about the San Francisco Estuary watershed is NOAA and the California Fish Tracking Society, Dr. Peter Klimley and Bruce McFarland, came up with this brainchild a few years ago about having a receiver array that spanned the whole entire watershed below the dams. And unfortunately, due to funding constraints, a lot of it has been dismantled. But during my study, you can see how diverse it was through the system, and this isn't even all of them that we had. So each one of these triangles represents a VEMCO receiver. And each time a fish swims by these, as Jacob McQuirk explained, the timestamp and the fish's number is recorded. These are transmitters that I implanted into my subadult striped bass, and specifically I used this one, which was a V9. And we looked at what presence was. Presence was calculated each time that a subadult striped bass swam past a receiver. Even though if it hung out there for a lengthy time, we took its arrival as one data point, so we didn't have to deal with pseudoreplication statistically. From this study, the 99 fish that we tagged, we ended up with records of 79 fish, which generated over 1.5 million data points. And we had visits to 256 receivers. So what I'm going to be talking about today is some products that we developed that are also in the manuscript for distribution, habitat use, and movement and residence. So this is a little bit about seasonal distribution. So you can see that the circles here are color-coded, And this was one of the very first products that we made because we just wanted to see where are subadult striped bass. So if you look at the yellow circles, you can see that during the summer, they're pretty much distributed from right above the confluence with the American River all the way down to the bottom of the Central Bay. During the fall, the population actually, or the tag population, actually constricts a little bit and shifts west, which is even more constricted in the wintertime. Then during the spring, we have fish that go out the Golden Gate into the Pacific Ocean, and we have fish that migrate all the way up into the feather and not upper reaches, but the upper part of the Sacramento River up to Colusa. And this is another view of that same data, but just looking at regional location. And kind of the take-home message here, when we're looking about predation, is that the Central Bay, Carquinez, the Central Delta, the Sacramento, and the North Delta are all areas that have fish or subadult striped bass present in them across all seasons. So those could be predatory hotspots at one point or another. But I think one of the other important things to see is over winter, the fish, we don't really see very many fish up above Carquinez Straits. And this might be an anomaly in that the South Delta had very few receivers deployed during my study. And we all kind of believe, or we know because we've been there catching these fish, that Clifton Food Court is heavily used during the wintertime. And so before we could actually look at habitat use at all, we had to map out salinity and temperature. And one of the things that's also been a little frustrating to me working here in the San Francisco estuary is everybody has their own definitions of how to define low salinity zone, what's an estuary, tidal influence, that sort of thing. So I was lucky enough to stumble across NOAA's new coastal and marine ecological classification scheme, which they developed specifically for the San Francisco estuary, that uses these exact same breakpoints, which are internationally accepted and peer-reviewed for the whole United States. And I think that using this side of scheme, when you're looking at introduced fish such as striped bass, it would be important to use these sorts of schemes and classification systems because they're used on the East Coast as well. So one of the things that we want to look at here is just, this is a typical Venice 1951 salinity level. So it goes from limetic to euhaline, or it goes from freshwater to marine. The breakpoints during different points per thousand, parts per thousand, I'll be talking about when we get there. They break the temperature into 4 different areas, starting with less than 10 in cold to warm of 20 to 25. And then they define channels much differently than the California Department of Fish and Wildlife does in the Delta. And they say that channels are greater than 4 feet and shoals are less than 4. So beginning here with the salinity use, I just want to have you take a look. The ocean is always euhaline, so is mostly the Central Bay. Up in here we have polyhaline, and here we start to get into ogliohaline, but right here, this whole entire area is basically fresh. And you can see that across all of the seasons. The other thing is, of course, the ocean stays euhaline the whole entire time as well. So when we're looking at fish abundance in terms of salinity use, where are these fish? So in the summer of 2010, the highest abundance was up here in the limnetic area of the watershed, although we did have abundance in mesohaline and in the euhaline areas. And then over the fall, we see sort of a segregation of the subadult striped bass in that here we have a bunch of fish remaining in the area that was limnetic, and then also, again, seeing fish hanging around in the central euhaline section, and then also the polyhaline. And in winter, we had very few to no detections, which was pretty interesting. So you can see here we have 1 detection, 2 detections. These fish are likely overwintering in the South Delta, which is our guess, but I maybe had 12 to 15 detections on each one of these fish. Was super limited to even say anything. But the majority of the fish shifted down and were in the San Pablo area or in the Central Bay, and then late in the winter they immigrated out to the ocean, and in the spring we had fish going out and coming back in, and some of the fish you can see were here utilizing all of the limnetic or freshwater section up here, and I'm thinking those might have been some male fish that actually went through puberty and engaged in a first spawning run. So in terms of water temperature, You can see here that this area predominantly is temperate, and temperate being 10 to 15, or 15 to 20 degrees. And then really the section up here is pretty warm during the summertime, or 20 to 25. And you can see how high the abundance is in the warm area, which really makes sense. I mean, these are pretty warm water temperate fish from the East Coast. And over here in the fall, we can see that there's fish using the cool area, which is 10 to 15 degrees, and then also the temperate area. And the most interesting thing starts happening in winter, where we lose all those detections. We don't really know where all the fish go. But what we do know is that right here, and this is the Sacramento River here, you can see the big pocket, and this is the Sacramento Ship Channel. It is cold, so it's 10 degrees and less is the average temperature over the winter time, and we don't have any fish detected in any of this cold area, in this water temperature, but we do have fish down here in the cool area of 10 to 15 degrees. The other thing I think is kind of interesting is during this time, the watershed is colder or just as cool as the ocean, which makes sense about why the fish are actually migrating during the winter. And then lastly, in spring, we can see that these are still the same temperature, which explains why the fish are going out and coming back in. And then spring of 2011 was actually sort of a hydrological anomaly. We had a lot of rain around May 5th and then again during Memorial Day, which really changed the way that the fish were moving. Even though it's the first time we looked at them, we saw them starting to move up as the water began to warm, and then they were actually moved back down or were flushed down with high flows. So looking at channel use, and across all seasons, the fish preferred, or they utilized the shoals much more often than channels. The only time that it wasn't really different was during the wintertime when they used 51% shoals, 49% channels. And I think kind of like the take-home message, we're still looking at this data a little bit more and then aligning it with turbidity by geographical location. The things that are kind of interesting to me is if you look at Carquinus, it's pretty much the same use that the fish are using channels and shoals in the same way, each across all seasons. So that I found kind of interesting. The other thing is that the fish are using, like Sacramento and the North Delta, even though that changes, their habitat use changes on whether they're using shoals or channels, that might have something to do with water temperature and or predation. For example, in the Sacramento, they use channels and shoals in the summer, but they're only using shoals during the winter time. And then in the winter, they're using more channels, which might be indicative of the higher number of detections being detected below the confluence with the Sacramento Ship Channel and the Sacramento River. And then in the spring in the Sacramento River, you can see they start using some of the channel, but majority is shoal use. And so the other thing that we really wanted to do, we started looking at the segregation of fish by salinity. We thought, wow, we wonder what's really going on with the individual fish. So we went ahead and we segregated all of the fish transects out for fish that we had 2 or more seasons on, so we could indicate which direction of their movement. And so we defined a movement as a relocation from one receiver to another receiver. Then we looked at all the fish, and there was a pattern in that fish that were exhibiting residence, it was residence by salinity. So the fish that were moving from one area to another were all pretty much following each other, and I don't want to call it schooling, but it was somewhat similar to that. So then I went ahead and mapped it using Esri's Tracker Analyst and set it to a time slider using the time slider extension. And then I mapped all the visualizations of all of the fish and looked at each one and lumped them to see which ones were most alike. And they fit a really nice pattern that we statistically analyzed. And then we went ahead and made visualizations in Camtasia. And this isn't as cool as the stuff that Jacob showed you, because I didn't do it in Eon Fusion, but it's what I had. So I'm going to show you the maps first, and then we're going to watch the video. So again, this is the low, we're calling this the Bay Area, this we're calling the low salinity zone, and this we're calling the riverine habitat. So the bay residents, which we had a total of 47 fish, we had 10 bay residents, 10 low salinity, and then the remaining fish were all in the riverine. And I may have biased because of where I tagged my fish to more highly select for riverine fish over the other 2 resident patterns that we see. but I don't know because we didn't tag fish any lower down, or I'm not going to have the opportunity to do so. So during tagging in the summer of 2010, the fish were up here in freshwater and then moved back down and pretty much stayed the whole entire summer in polyhaline or the bay area. And then during fall, they were moving back and forth, and we ran a couple little statistical analysis with the movement, and it did seem like it was related to the tide. But I haven't finished up with that either. And then during the winter time, the fish moved back and forth, basically what we think is, again, following the tide. And then in the springtime, they dropped down and exited through the Golden Gate Bridge, and then they came back during the summertime. These fish, during this whole entire time, after they moved out of freshwater, were either in polyhaline or euhaline water. So we're calling those bay fish or high salinity fish. On the other hand, we had the low salinity zone fish, which actually moved between the lower reach of the Napa River and back up to right above Decker Island, back and forth with the tidal surge. And this salinity there was basically ogliohaline, or it was always, like 3 to 5 parts per thousand. So it was ogliohaline or mesohaline. And then in the springtime, they moved down a little bit more with the freshets that came down. And then in the fall, in the fall, they moved back up and pretty much were doing the same sort of movement patterns that we saw during that same type of year. I think the most interesting fish, and the fish that would have some bearing on looking at predation as a whole on smolts, are basically the riverine fish. And they followed this pattern like through the whole 17 months that we studied them. So in the summertime, they stayed up here in freshwater. When the water temperatures began to drop off, they came down the Sacramento through the Delta Cross Channel into the Mokelumne and into the San Joaquin and down to the Clifton Food Court and hung out down here, where we lost them. And we lost them because, again, we didn't have any detection equipment, and the detection equipment that we did have, we lost, and it was unsafe for us to get it back. But if we get it back, I would love to look at it at some point. So we lost them for the whole entire winter. Then in spring, when the water temperature started to rise and to about 14 C, they turned around and left through Old Middle River, through the San Joaquin, up the Mokelumne, through the ship channel, and then back up to where we saw them the previous year. So I'm going to show you the visualization analysis now, and so they run pretty quickly. But I can stop them if you'd like me to, and the panel is definitely going to have access to looking at them after my talk. But what I want you to follow basically is follow the bouncing colors as they move down the stream. Okay, so here we have the fish post-tagging moving down during the summer into the Central Bay. And then fall comes up, and they're back in that area, same as well. And all of this is polyhaline, it's the fall. And during the winter, they move up a little bit farther and then back down again. And what you're seeing is the change in salinity between rain and no rain during that time. And then during the spring, they move up again in that area, and then in a few minutes, we'll see them drop back down and exit the Golden Gate, and then return later on. So this is a low salinity zone fish, and you can see the movement. Their movement patterns are different, and they're utilizing water of different salinities. And then they're down during the late fall in the same area, and during the winter they move back up, which is exactly the opposite movement patterns that we see with the bay residents. And then we believe that during some of the winter freshets of 2011 that they were just pushed down, and if you look at the salinity gradient, it's mesohaline. And these are the most interesting fish, I think, are the river fish. And here they come in the summer, spending their time up near the confluence of the American River. And then during fall, they go down to Clifton Court, where we lose them. And we're not having any data right now because we have no data right now. And then during spring, you can see them start on their way back up. And then we had a big rainstorm, which washed them back down. This is like the end of May, and then we watched them go back up again. And this is just a group of fish that acted like low salinity zone fish. And this isn't part of the manuscript, but I thought it was really interesting. Because these fish act like the low salinity zone fish, but during the spring, we get to the spring, we're in the fall now. But during the spring, they actually end up migrating all the way down to the South Bay down in this area here and actually move up Coyote Creek. And there was a graduate student from Moss Landing whose name is Mike Walsh, and he actually did otolith studies of the fish, and he found the same pattern that I found with these residents as this other fish, that he had otoliths that matched fish that were collected down in the South Bay. So our data kind of hooked up, serendipitously. It was pretty neat. So just the conclusions of our study. Basically, what we know about striped bass because of the study is that they're widely distributed until the early fall or late fall and the early winter, that there is a shift towards the ocean when water temperatures decrease, that the seasonal abundance is highest in limnetic and polyhaline habitats, that we know that abundance as a whole is highest over 20 degrees C, and fish are not observed less than 20 or 10 degrees Celsius. That it appears that shoals over channels across seasons are used, and that so far, based upon the data that we have, which is one 17-month telemetry study, that there appears to be bay, low salinity zone, and riverine residence patterns. And so some of the things I think that would be really important and interesting to do is we have a plethora of telemetry data that IEP has funded and other studies, and I think it'd be really interesting to synthesize all of the telemetry data along with the trawl and seine data sets to determine season and regional habitat overlap between predators and prey. And I'm going to show you a real rough shot of a product that I presented at the Salmonid Restoration Federation a couple years ago. One of the most important things, if you really look at smolt out-migration timing and overlay that with the tracks of my subadult striped bass, you will see there's not a whole lot of overlap. So you get down below Sherman Island, which is, down into Carquinez Strait. Then I think there might be predation issues going on there. I think it'd be really important to look at birds, river otter, seals, sea lions, in terms of other predators on out-migrating smolts besides striped bass, because they're likely also in the ocean. And that's not saying they're not predating on smolts in the ocean, because I believe they probably are. And then continue to incorporate deterrents and attractants to shield smolts from predatory hotspots. This seems to me like a really logical next step since the watershed's so highly managed already. And in closing, I'm just going to show you this this figure, and I apologize because it was looking kind of cloudy. So this is July 2011, and these bars right here show the gray bar is the number of detections, and this little tiny black bar is how many striped bass were actually there. And then you can see the same thing here, and this is how the number of detections in relationship to little number of detections, number of fish. So what we also did was we mapped in a wheel using the log sum of all of the different species that were present at that location at that time from data that was collected in a trawl or in a seine. So if we want to look at smolt tagged striped bass overlap, you can see that my striped bass were here right below the confluence of the American River, and lo and behold, there was a salmonid smolt there. I don't know if that smolt is a Chinook, but I would guess because of the timing that it's definitely a steelhead, and I lumped them together for this very quick exercise. And we have these for each month, but this is the only one that I could really show smolt and subadult striped bass overlap, so I thought I would show you the evidence, I guess. And then right here in Carquinez Strait, you can see that we have smolts again, and that we have striped bass not only caught in, we have co-occurrence of striped bass caught in the traps or trawls or seines, but we also have them from my biotelemetry study. And then down here in the bay, we're not seeing any. So my question is, I wonder, are any of these smolts even making it out of here down in this area during this time? And if you would like to contact me, you can contact me at my UC Davis email or at my Fish and Wildlife email. And thank you again.
Maybe just a quick clarifying one. Sure. At the beginning, you said you sampled hundreds, but tagged 99. What were the characteristic size of those ones you actually tagged? Yeah,
the fish were, the mean fork length size was 285 millimeters. The 99 fish that I tagged went from 225 to 410. And I'm those fish were worth a lot of money by the time we, captured them, tagged them. So the fish that I didn't tag were either had an external parasite or looked to be too thin, or a lot of them had, a fungus on them. So we just selected to only tag the most healthy-looking fish.
For your habitat use data, you had that channel shoal separation. I'm wondering if those data are from a situation where you have, in the same spot, you have shoal and channel, and you have a sensor in the shoal and the channel. So as a consequence, if you say they're in shoal, you know they're avoiding the channel. Or whether they're just, your detections are, well, that was a channel sensor, we saw it there, and shoal is, we had a shoal sensor and we saw it there.
And you know what, I apologize, I should have clarified how that data was attributed. So the Department of Water Resources and USGS has amazing bathymetric layers. So I basically brought those bathymetric layers into Google Earth and then used latitude and longitude on the receivers to place them where I thought where they were on that, and it was really clear about where the receivers were deployed, if it was a channel or if it was a shoal. And then we have metadata analysis that the Fish Tracking Consortium had that actually showed or tested how deep of water that receiver was placed in, so I used that. And when it couldn't make sense or it looked like the data could be erroneous, I didn't use it. Yeah, so I can't tell about avoidance one way or another. They were either present or not present, or not observed.
What was your definition of a shoal in Carquinez Strait? My understanding is that's a pretty deep channel and not really has a shoal.
Yeah, they're actually in Carquinez Strait. I was defining Carquinez Strait from the confluence with San Pablo Bay all the way up to Sherman Island. So that whole area is what I was calling Carquinez Strait, based on salinity gradient and water temperature. So where the shoals are there are in and around Sherman Island, where the mothball fleet is. Yeah, so it's really clear. And then the other way that
So it's really Sassoon Bay and Carquinez Strait, not necessarily.
Or it's the shoal at Carquinez Bridge. So there's a very definite shoal on either side of the bridge, and from the bath layers, it's very clear to see where they're landed. And there is actually kind of a good-sized shoal there. I mean, it's nothing like other areas, but you can relatively clearly see where the dredge marks are in relationship to where the receivers were placed.
Thank you.
Just can I ask one real quick question? What is the detection range for these particular receivers?
Great question. It kind of depends on the noise. Usually it can go anywhere from 80, 100, 200 feet in places that are really quiet. But what we do, for instance, underneath bridges, there will be literally sometimes 50 to 80 receivers deployed underneath, like, say, the Golden Gate Bridge. So we have the best possible benefit of collecting them. But where it's very quiet, it's very long. It's not as long as JSATs, but it's actually location dependent on receiver capability. So I would have to know more of like a hertz, like it.
Okay, good.
All right.
You'll be around for more questions.
Yes, absolutely. Thank you so
Excellent.
much.